Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Mariam Aisyah binti Muhammad Khairul Fuad and my number matrix AS11701. Today, I will present under the subtopic 28.1 Evolution of Animals. So at the end of this subtopic, you must be able to list 3 common characteristics of animals that are not found in other eukaryotes. Second, summarize the colonial flagellates hypothesis as it relates to the origin of animals. Third, distinguish among the different body plans of animals. And the last one, compare protostome and deuterostome development. Now we go through to the characteristic of animals. First, all animals are multicellular. It means that animals are organisms that made up of many cells. And second, animals are heterotrophic, which means they must acquire nutrients from an external source. Okay, animals are different from fungi because animals ingest the whole food and digest it internally, but fungi digest their food externally and absorb nutrients. Okay, second, muscle and tissue allow motility and variety of flexible movement. Motility enables animals to search for food and prey on other organisms. This movement also allows animals to seek for mates, shelter, and suitable climate. Okay, next, many reproduce sexually, some asexually, and some combine both life cycles. So we can see here that animals have a variety of life cycles. And animals undergo a series of development stages to produce an organism that have a specialized tissues that carry on a specific function. And the last one, animals are descended from a single common ancestor. Within the animal lineage are 2 main branches, invertebrates and vertebrates. As we know, invertebrates are animals that do not have a backbone, and vertebrates are animals that at some stage of their life have a backbone. So for summarize, the common features of animals are multicellular and animal cells lack a cell wall, which common to both fungi and plants. Although the cell wall in fungi and plants are made from different organic molecules. Okay, then animals are heterotrophs, animals are typically motile, and most animals reproduce sexually. So I think that's all from me. Thank you, and Fakri will continue their presentation.
Bismillahirrahmanirrahim. Assalamualaikum and very pleasant day to all of you. My name is Muhammad Fakri bin Zulkifli and I'm going to explain about the ancestry of animals. Before I explain all these 5, let's take a look at the title first. Ancestry of animals. So what does it mean by ancestry? The root word is ancestor, meaning origin, basic, fundamental of a thing, or the pioneer of a thing. So basically, ancestry of animal is the origins of animals, where it coming from, how the animal evolved from something to something. For your information, the evolutions of animals is actually based on the colonial flagellate hypothesis. In the next slide, I will give the visual explanations about the hypothesis. The hypothesis actually explains the process, how the multicellular organism evolved from a single flagellated cells. Scientists believe that the combinations of these unicellular flagellates will forms aggregate before it forms holosphere structure, and it will undergo some specialization of cells before it turns into tissues. As you can see in the first box, the hypothesis states that animals are descended from an ancestor resembling a holospherical colony of flagellated cells. Next, among protists, the conoflagellates most resemble the last single-cell ancestor of animals. These conoflagellates is actually types of the unicellular organisms. Movements of flagellums creates water currents. Conoflagellates can also exist as a colony of cells. And why is hypothesis so popular? This hypothesis is attractive because of its implications regarding animal symmetry. This is what I call as colonial flagellate hypothesis. As you can see, there are a few stages here. From a single flagellate, it will turn into aggregates and it will form into a holosphere. This is the cross sections of holosphere. There's a space there, and some of the flagellates will undergo specialization of cells, into reproductive cells like sperm cells. And next, it will fold in and creates tissue, as you can see. Looks how this hypothesis states from a single flagellate and returns into tissues. The evolutions of body plan. Diversified of body plans characteristics of animals that we can see nowadays is actually present by the Cambrian period or Cambrian explosion. It can be distinguished by different combinations of numbers, positions, sizes, and patterns of body parts. This differentiation combination could have led to the great variety of animal forms in the past and present. Evolutions of animals can be divided into two, symmetry and embryonic development. It's still under the same topic of phylogenetic tree of animals. Symmetry, a pattern of similarity that is observed in an object. So there are three types of symmetry. We have asymmetry, radial symmetry, and bilateral symmetry. So what does it mean by asymmetry? Asymmetry is actually something that does not even have symmetry, or I can say it's not have a particular shape. It's not even symmetry. Okay, you get the idea, right? Next, the radial symmetry, something which has a cylindrical shapes. As you can see here at the top of the slides, radial symmetry. There's some symmetry parts here, and the center at the middle of the thing. So examples of this radial symmetry except of this is actually the starfish, the jellyfish, anything which have a cylindrical shapes and the centers of symmetry. Bilateral symmetry is something which has couples of symmetry. As you can see in these turtles, there's symmetry from the side view, there's symmetry from the front view, downside view, which means something which has variety of many angles of view and have some or once or more symmetry of it. As you can see, they have a ventral, dorsal. These turtles have symmetry parts from the left side and the right side, from the top side and the bottom side. Next, the embryonic development. Scientists say that the first tissue layers that appear are called germ layer. There are two types of tissues layer, which is diploblastic and triploblastic. So what is the difference between these two things? Diploblastic, diploid. So it have two layers of tissues, and three layers of tissue. That is easy for you to distinguish these two things. Diplo, two layers, triplo, three layers. These protostomes and deuterostomes will explain more by Nisha on her slide. But in rough, photostome is actually the first of embryonic opening development, and deuterostomes is actually the second embryonic opening development. So that is all from me. Thank you.
Assalamualaikum. My name is Khairuni Shawsabi Desmond. My matrix number is AS12337. And I will continue the presentation. So, protostomes versus deuterostomes. Proto means first, deuter means second, and stoma means mouth. So, protostomes means mouth first, and deuterostomes means mouth second. So, most invertebrates are protostomes, where mouth develops prior to the anus, and all vertebrates are deuterostomes, where anus develops prior to the mouth. The difference between protostomes and deuterostomes can be differentiated by three major events, which are cleavage, blastula formation, and coelom development. So the first event after fertilization is cleavage. So in protostomes, spiral cleavage occurs, and daughter cells um, grooves, and the daughter cells sit in grooves um, formed by the previous cleavage. So as we can see from the slide, the blue one is the daughter cells, and the daughter cells sits between two cells. So the fate of these cells is fixed and determinate, where it means that each can contribute to development in only one particular way, which is this is the only structure that the embryo can grow. So in deuterostomes, radial cleavage occurs, and the daughter cells sit right on top of the previous cells, as you can see in the picture. So the fate of these cells is indeterminate, where it's where if they are separated, each cell can develop to another organism. Next stage is blastula formation. The indentation produces an opening called blastopore right here. So in protostomes, the first opening becomes the mouth because the mouth appears at or near the blastopore. And in deuterostomes, the the first opening becomes the anus because the anus appear at or near the blastopore. The last stage is coelom development. So certain protostomes and all deuterostomes have a body cavity lined by mesoderm called coelom. So in protostomes, coelom forms by a splitting of the mesoderm, and in deuterostomes, the coelom forms by an outpocketing of primitive gut. So the gut is over here. Learning outcome for 28.2. The first one, explain why sponges are considered to be simplest animals. Number 2, discuss how a sponge respires, feeds, and reproduces. Number 3, compare the anatomical features of ctenophores to those of cnidarians such as hydras. Okay, these are some of the simplest invertebrates: sponges, comb jellies, cnidarians, sea anemones and corals, and hydra. So that is all from me, and I will pass the presentation to Haza.
Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Haja binti Jafrina and my metric number is AS11767. So, today I'm going to talk about sponges. Okay, as we know, sponges are aquatic, largely marine animals that vary greatly in size, shape, and color. Sponges are categorized in the phylum Porifera. So, we're going to talk about their characteristic. Okay, the first one we have, they are lack of true tissues. They have only a few cell types, which is they are also lack of nerve and muscle cells. They have sac-like bodies that are perforated by many pores. They all have a canal system of pores of varying complexity that allows water to move through their bodies. The interior of canal is lined with flagellated cells that resemble choanoflagellates. In a sponge, these cells are called collar cells. Okay. The beating of flagella produces water currents that flow through pores into central cavity and out of osculum. They also have sessile filter feeder. Okay, what is the function of filter feeder? The function is it filters suspended particles from the water by means of straining device. They also have the skeleton of a sponge to prevent their body from collapsing. All sponges contain tough fibers made of spongin. They also have the endoskeletons that contains spicules. Traditionally, the type of spicule has been used to classify sponges whether they are bony, glass, or spongin sponges. For the reproduction, they can produce both asexually and sexually. For the asexually, their reproduction by fragmentation or budding. Sponges are like the other organism. They are capable of regeneration or growth of a whole from a small part. Okay, we move to the next slide, which is we want to learn about comb jellies. Comb jellies, known as stenophora, they are usually found in a warm water. Okay, they are solitary, which is they are free-swimming marine invertebrates. Okay, stenophores, they are represent the largest animals that propel by beating of cilia. The third point is they are made up of a transparent jelly substance called mesoglia. As you can see in the picture, they are transparent, right? Okay. They are bioluminescent organisms that can produce their own light. They are also capture prey with chloroplasts. Okay. For the next point is, traditionally, stenophores were placed as a group with the cnidarian. However, recent studies suggest that comb jellies contain ancient DNA sequences, which is they are not in the same group with cnidarians. Okay, for this slide, we're going to learn about cnidarians. What is cnidarians? Cnidarian is a tubular or bell-shaped animal that often reside in shallow or coastal water. As you can see in the picture, they have tubular or bell shape. Okay, cnidarian also found in the freshwater, brackish and oceanic form. For the term of cnidaria is derived from the specialized stinging cell that called cnidocytes. Each cnidocyte has fluid-filled capsule called nematocyst. Okay, for the third point is they have two-layer body sac, which is outer layer and inner layer. For the outer layer, they are for protective epidermis. For the inner layer, they secrete juices into internal cavity. The fluid-filled gastrovascular cavity also serves as supportive hydrostatic skeleton. For the fourth point, they have two basic body, which is the polyp mouth is directed upward, and the mouth of a jellyfish, or we call as medusa, is directed downward.
Hello and good morning everyone. My name is Ahmad Nurhan Amshad bin Iza and my number matrix is AS12285. Today I will present to you about sea anemones and coral. First of all, sea anemones are sessile polyps that attach to submerged rocks, timbers and other substrate. Most of sea anemones range in size from 0.5 to 20 cm in length and 0.5 to 10 cm in diameter and often colorful. It has upward turned oral discs containing mouths in the center of this, by tentacles containing nematocytes. This is tentacles. For corals, corals resemble sea anemones encased in calcium carbonate. It lives in colonies and build coral reefs. They exhibit elaborate geometric designs and stunning colors. Next, I will explain about the simplest invertebrate, which is Hydra. Hydra is freshwater cnidarian. It is small and have tubular polybody, about 1/4 of an inch in length. It reproduces asexually and sexually. By asexually, it forming buds, small outgrowth that develop into a complete animal and then detach. For sexually, the interstitial cells of the epidermis are capable of becoming other type of cells such as an ovary and testis. When hydras reproduce sexually, sperm from a testis swim to an egg within an ovary. The embryo is encased within a hard, protective shell that allows it to survive until conditions are optimum for it to emerge and develop into a new polyp. Hydra contact with the nerve cell within the nerve nets. which present in epidemics. These interconnects nerve cells allow the transmission of impulse in several directions at once. It has 4 to 6 responding tentacles that contain a large number of nematocytes. Here, this is reproduced asexually, which is a bud, and then detached with the hydras. And lastly, the simple invertebrates through jellyfish. Through jellyfish, also known as sea jellies, the medusa is primary stage and polyp remains small. Jellyfish are zooplanktons and depend on tides and currents for their primary means of movement. They feed on a variety of invertebrates and fish and are themselves food for marine animals. So this is medusa fish and this is polyp fish. That's all from me. Thank you.